Hello, my name is Victor Huber, and I am at the University of South Dakota. Today I'll be presenting the background for our project that we're working on, looking at the detection of antibodies against SARS-CoV-2 in cancer patients. This project is being done in collaboration with two clinical faculty, Santiago Lopez and Fernando Bularudis, who are at Sanford Children's Hospital, and another basic scientist in the Division of Basic Biomedical Sciences at the University of South Dakota, Khosrow Rezvani. Overall, we're looking at the impact that SARS-CoV-2 infection has on patients with that have been diagnosed with cancer, using two major specific aims. One, determining SARS-CoV-2 antibodies in cancer patients, and two, looking at the impact of SARS-CoV-2 immunity on biomarker expression in these cancer patients. The main comparator is going to be understanding how the interaction with the immune system is different in patients that have been diagnosed with cancer versus patients that have not been diagnosed with cancer. So to do this initial study, we'll be looking at antibodies in the serum of patients that have been infected with SARS-CoV-2. We'll use antigens, specifically the receptor binding domain protein of SARS-CoV-2 and the full-length spike protein of SARS-CoV-2, and then interact antibodies from the sera with these proteins, and then use the ELISA assay to tell us whether or not we can detect antibodies against SARS-CoV-2 in these patients. We'll look at different antibody isotypes, things like IgG1, IgG2, IgG3, and IgG4. We'll also look at the IgA that's in circulation, as well as IgM, as ways to identify the sort of breadth of the immune response against SARS-CoV-2, looking at the different antibody isotypes that are known to exist in circulation. Once we have determined how these antibody profiles look, we'll move forward with a neutralization assay. This neutralization assay uses a pseudovirus that will interact with cells that are expressing the ACE2 receptor for SARS-CoV-2, and the pseudovirus expresses the spike protein of SARS-CoV-2. The pseudovirus can bind to ACE2 and infect the cells or get taken up by the cells, and once it's taken up, it can provide a fluorescent signal that can be measured that will indicate the ability of the virus to actually get into the cell. Presence of antibodies would block that interaction, and thus a positive result from this assay would be that fluorescence would not be seen in cells where the antibody effectively neutralized the pseudovirus. Finally, since neutralization is not the only part of an antibody that plays a role in protection against an infection, Fc receptor interactions inducing things like antibody-dependent cellular cytotoxicity or antibody-dependent phagocytosis are being evaluated using an ADCC assay, specifically the Promega reporter-based ADCC assay, which uses a luciferase reporter expressed by Jurkat cells that have Fc receptors on their surface to indicate a positive interaction. So in this case, we'll use proteins or target cells expressing the SARS-CoV-2 spike proteins, add antibodies from the patient samples, incubate those with the target, and then bring in the engineered Jurkat cells. If they engage those Fc receptors, there will be a luciferase signal provided that will be measured and will indicate Fc receptor engagement and activation. The goal of this is to give us an overall view of how the immune system is recognizing the virus as part of that next exposure to the virus in these patients, and to determine whether the current immune environment would be protective, or if additional interventions such as vaccines or other sorts of therapies would be needed to help patients that may not have fully responded against this virus upon first exposure. In aim 2, we're going to be looking at the contribution of SARS-CoV-2 immunity toward biomarker expression in cancer patients. We'll be using biomarker assays that will allow us to understand how the immune system has affected the expression of specific biomarkers that are part of a bank of biomarkers that Dr. Rizvani has been working with in studying cancer patients and understanding these responses in these individuals. Overall, in addition to understanding the immune response against SARS-CoV-2, we'll also be understanding how that immune system response affects biomarker expression within these patients so that we can understand, again, what interventions and what sort of future treatments and vaccinations are needed for these patients that have been exposed to SARS-CoV-2. This will help guide future vaccine administration in cancer patients and our understanding of the immune response in these individuals. Thank you.